Hello, everyone. I hope your weekend was fine. It's very glad to see everyone, and we almost at our at the end of our class. We are now in lecture number 7, and I am Dr. Hadar Ben-Yoav from the Ben-Gurion University of the Negev. And today, we're going to learn about how to utilize all this kind of equations or physics that we learned into designing and simulating a lab on a chip. And we're also going to learn some advanced modeling techniques to decipher the signal, the output signal that we measure from the lab on a chip, that we will understand from the signal how it's correlated to the analyte that we have in the sample. Just a second. Here, okay. So there are different types of simulations, modeling. We know the theoretical calculations that provides us mathematical solution. And there are sometimes that we would prefer to do numerical simulations when, for example, we cannot solve the equations, or even when we would like to know some complex interactions between multiple equations or physics. So today, we're going to the first part of the modeling. We're going to describe some key parameters. What should you do when you want to build a good simulation of your lab on a chip? And then we're going to study how we can, what kind of calculations or what kind of parameter we can use to analyze the performance of the simulation that we built. So how to build a simulation? The first thing that you want to do when you want to build a simulation is not touching the computer. You need to ask yourself two main questions. Do we really need a simulation? I mean, it's very cool, and we show some equations and we solve the mathematics, or we show some very nice figures, but do we really need a simulation? Because not always we should do a simulation when we don't exactly really need for that to understand about the system. Okay, we do a simulation when performing an experiments would cost a lot of money, so we want to save some money, or if actually testing specific parameters of the system are unable doing experiment, we cannot do that through experimental approaches or techniques, and we need to do that theoretically. And then we need to ask ourselves, okay, so we said we came into conclusion that we need a simulation. Now, what is the problem? What are the physical mechanisms? Oh, I lost you. Second. Yeah. What is the problem? Okay, what exactly we want to get from the simulation, that what exactly we don't know that we need a simulation to solve for us, to know the solution. And then what are the physical mechanisms? For example, we talked about the diffusion or the electron transfer rate in the last couple of lectures. Now, not always when we build a simulation, we are going directly for the 3D nice models. There are nice pictures, but we don't always need it. And actually, it's even better if we can reduce the dimensions of the model. So what is the geometry that we will need? Is it a direct line, direct channel? Or maybe it's only the surface of an electrode? Or can we reduce its dimensions from 2D or 1D? For example, if we need an electrode surface, which is radial. So do we need all the, well, it's kind of like, do we, if it's radial coordination, do we really need all the three different radial coordinations? So can we reduce it into only the radius and the angle, for example? And then the next step is to use or define the boundary conditions, such as if it's a flow in the solution in a channel, where the walls, where the boundaries of the solution of the flow, and you want to model the flow, so where the walls are. And eventually, we want to define an appropriate mesh, and I will explain later what is a mesh. So the better the planning, the better the simulation. Before we even touch the computer, we should answer all these kind of questions, and we will be able to, and then we can approach and go into the building the model. And believe me, I've been there. You don't want to do multiple things. You want to be as much as efficient as possible. Do we have any questions on this slide? Great. So let's look at numerical models. So numerical models, it's a model that we don't have the theoretical calculations. They are built on some kind of physics, differential equations. So we are building the geometry of what we want to solve, such as, for example, let's take an example of the microchannel. Sorry, we're adding the physical parameters and dynamics. We talked about what is the physics, what is the mass transfer, what are in a channel we have the dynamics of the flow. Then we apply a numerical solution. We solve all the model itself with the boundary conditions and so forth. And from the solution, we extract the actual information that we need. So if, for example, we solve the flow in a channel, and we actually wanted to know the flow, which was 10 microns far away from the channel walls, then the analysis is extracting the actual velocity at the 10 microns from the wall. We do visualization, if it's either plots or figures, and eventually exporting the data. So what is the first step when you go into building the simulation? The first step is, after we define, of course, all the answers to the questions, to the planning, the first step that we do when we go in building a simulation is, should we use even a computer, or should we do actual calculations on a paper? So, but it's been, do we really need numerical calculations, or can we actually use theoretical calculations that actually can give us the solution in an analytical way, mathematical way? And usually, in most chances, we would prefer using the calculations, the theoretical calculations, because if you look at this kind of range from a concept into reality, and you want to advance us from actual idea to how it's going to look at reality, theoretical estimates give us very good estimation for how reality is going to look like. And the efforts are kind of minimal. We don't need to use any strong computers, software. We actually just need a paper and pen. And when we look at numerical models, yes, they will indeed add the estimation. There will be more. They will give a better estimation of the reality than theoretical. But again, it's only the addition. Some additions, which, you know, even less than 10% addition, that not always necessary. So I always want to emphasize checking if you can solve your problem of building the simulation based, the model based on theoretical estimations. Do you have any questions on this slide? Okay. Oops. So here I took from the book some comparison, pros and cons, advantages and disadvantages for using theoretical calculations versus numerical models. And here, for example, the preparation, when you want to prepare is for the to build the model. In theoretical model, it's kind of a plus because you really need to learn the theory. You really need to understand theory, which provides you better insight into the whole system. While numerical simulation, it's kind of a software modeling guide. So you actually just need to put, its tutorial gives you exactly where to put all the parameters and you run it. So you don't really know what exactly parameters you really need to add or to in order to get a more accurate estimation. Output. In theoretical models, you have a minus here because you get a formulation, you get the equation, you get the result, and it's not very, let's say, sexy. It's very dry, it's numbers, it's not exactly a nice visualization. And here the numerical simulation has the plus because it gives very nice 3D illustrations, provide a very increase the appearance of your data. Applicability range. The good thing is the theoretical model have a plus because it gives you a kind of a range of solutions. You don't need to really solve it for a specific, solution. It gives you an equation that shows how the result will behave for a range of parameters. Such as if you look at the flow, so we can get the solution for different locations as a function of time, as a function of location. So we just need to put the different locations and we can see the trend. While numerical one gives you only a specific solution at a time or specifically what you solve the structure or the specific condition of your system. Insight into physics, 2 pluses, because usually it's used dimensionless numbers. So you can get understanding about exactly how the physics behaves, such as a Peclet number. And also for numerical, we have a plus. Also can give us more information about the physics, how it behaves, how the geometry affects it, the solution, for example. Implementation time. For the theoretical, it's fast, eventually you apply the solution. It's sometimes hard to get the solution, but it's kind of fast. You don't really need a lot of calculation time. While for the numerical, it's a minus because, and it's a very big minus, because you need a lot of time to build a model, then to correct it maybe if you did some mistakes, and then to apply the simulation, the software, and you need maybe to change it because maybe some parameters are wrong, and so forth. And also you need very strong computers, and the simulation can run sometimes even a day or two. So all of that is a big minus because it still takes time. Now, so these are the main comparison, and there's no straight answer if it's either good based on this. You always need to get more experience in using models. So when we look at this kind of range from the concept to reality, another important thing other than theoretical and numerical model, and this is a very big minus for numerical models, is that sometimes, and not sometimes, actually I saw that in a lot of examples with students, that they use the numerical model, they build the model, they solve the simulation, and they get some amazing results, which actually doesn't even make sense. And this is a big disadvantage, that you need to be very much aware of bad numerical models. Bad, I mean that they solve it, but they give you the wrong answer. I will show an example for that. But let's be after talking about the mesh. So when we use a numerical model, we have something that's called mesh. Usually, we use models or software based on finite element modeling, which kind of mesh all your space into small structures, such as here it's example of triangles, sometimes even different other types of mesh. And then it solves the equation for each one of the triangles in the mesh. Now, a good mesh meaning that you have big parts or mesh where you don't really expect to have any big changes or any big changes that affect the simulation, and very dense mesh where the area that you really think is going to change dramatically. For example, if you look at this kind of similar mesh everywhere throughout the space, we have some solutions that, although it's going to look like this, but actually all of that solve the same. So we are going to actually miss this kind of small reactions happening at the smaller dimensions. Now, if we use this kind of mesh, we're gonna catch the actual changes in this small dimension, and therefore we're gonna have a much higher much denser mesh than in places that we don't have a big change. So it's very important to have a good mesh that we shouldn't have, very dense everywhere, then that's mesh everywhere, because then the calculation takes years. We actually need to find or use our common sense and know where the changes should be very very small, very high resolution. Then over there we should put the high mesh. In places that we don't have a big variation, we should use the less denser mesh. So bad mesh can not only provide the wrong solution, but also actually can provide no solution at all, because calculations. And I saw a lot of simulations, and you need to, there's a whole theory on that, on using good mesh and papers, how to overcome those kind of challenges. Any questions on the mesh? Okay. So this is an example. So the more elements we have, the better solution we get. Let's look if we actually, if we get more elements, do we get a better solution? So this is an example of a channel with a length of 200 microns and a height of 20 microns. And here we can estimate, from theoretical calculation, we can estimate, first, we can reduce the dimensions of the theory from 3D channel to 2D geometry, because for us, it doesn't really matter, what is the width. It's going to be the same everywhere. And we can expect that to use calculations for the flow rate, which is 2D flow rate will be 5.556 10 to the minus 6 meters squared per centimeter. So we look at the flow rate. It flows through the channel with a height of 20 microns, and the width, basically, we divide it by the width, and we get, this is the flow rate that we get, a 2D flow rate. Now we built a simulation, and this is a result from a paper that showed that if this is the theoretical, the one that we calculated, now we need to estimate the flow rate with a numerical model. And this plot shows the calculated flow rate, okay, divided by the, or it's a function of the number of mesh elements. So the higher the number, the more elements or more denser mesh we use. And what you can see that if we use low number of mesh elements, we get some kind of accuracy, about 75% of the flow rate, the theoretical flow rate. However, when we increase the mesh elements, the number of mesh elements, we get a solution which is more and more accurate. However, at some point, it doesn't matter how much we increase the mesh, number of elements in the mesh, we don't really change the solution, we just add the more complication to the model, to the calculation numerical model. So a good place to work is around 95, 98% accuracy. And this, before you are, or during your build of the simulation, we want to test for different mesh elements and to find where is the most efficient number of mesh elements that we want to be. Any questions for this slide? Okay. So let's see another example for the importance of the good mesh. And here we have a channel, some flow, some liquid fluid flows in the channel. And we have this kind of areas, two different areas, where the there is some kind of flow of solution towards this kind of end. So we have a kind of like open channels at both sides. So flow is flowing here and also supposed to flow from here. Now, the first part that we need, we can improve the mesh in expected areas. Sorry, the first thing that we actually see that it's a symmetrical problem. Similar solution of the diffusion gonna be similar as here and as in here. So whatever we solve here, it will be similar to here. So we have a symmetrical solution. Let's improve the mesh in the expected, in the expected areas. So we expect to have more, let's say, different solution or changes in the areas where we have the diffusion or the channels that go inside from the side. But accidentally, we only improve the mesh on one side. And we had a lower mesh or less denser mesh in the other side. Let's see what happens. And this is the concentration as a function of the y coordinates, meaning the concentrations. This is the more higher concentrations is in red. So the concentration from here to here. And again, we look about diffusion. So some material concentration is supposed to come from the sides inside and from the side inside. So when we use a not symmetrical mesh, a wrong mesh, what we see, is that, okay, we have high concentration on the side because this is where the concentration arrives on one side. We have the high concentration, and then it goes down into minimum because this is the middle of the channel. And then we have something weird on the other side here of the channel, you have something weird, which actually, if you see this is 0, it actually goes into minus, negative concentrations are observed. So this is when I mean that when we have a wrong mesh, we actually solve the model and we get bad numerical results because we don't have such thing as negative concentration. Okay, so get the results. Don't believe it always, and always question yourself, does it make sense? Do you have any questions on this slide? So when we choose the correct mesh, and we have high diffusion from here, high diffusion from here, and we look at the cross section, what we get is a high concentration on one side, high concentration on another side. And this is the cross section. Sorry. Here. So we have high concentration on one side, high concentration on another side. Again, after the diffusion, and 0 at the middle because all the material diffused outside. And this is a good model because we use the high density mesh in both sides of the channel. Does anyone have any questions on that? Okay. Now, there are different types of numerical modeling software. The most known one is Comsol Multiphysics. Very versatile software, very intuitive, very nice. You can actually integrate different types of physics. Very recommended for you to test. There's the Conventor, ANSYS, Flow3D. Some examples of numerical modeling papers or reviews that you can look for. I guess there are also other examples that summarize or compare the different types of software, and it's very good to do this kind of research before you choose the software that will give you the most optimal solution for your problem. For example, review on mixing in microfluidics, showing theoretical and experimental results for different modeling software. So if we summarize how to build a good model, the first thing that we do is we don't use a computer. We take a pen and a paper and do the initial calculations. Do the initial calculations, understand the system. Do we actually really need the model, and so forth. Then we look for symmetries, and we simplify the geometry. Then we define the initial and boundary conditions, input parameters, what kind of constants we should use. And eventually, a very important step is defining the required mesh. Remember, a denser mesh doesn't mean always it's a better mesh. It should be kind of in between, kind of a range of meshes, and also find the location that require the denser mesh. And the most important thing is GIGO. GIGO is garbage in, garbage out. Garbage in, garbage out, meaning that you get a result, you don't open a champagne yet, you look at the results and you try to see if they're making sense. Okay? Don't come with these kind of results, try to think they're making sense, because in a lot of examples, they're actually wrong. Okay? That's what it means, garbage in, garbage out. You put bad parameters and you get garbage, which is bad numerical models. And obviously, there are a lot of tutorials and demos on the web. Okay, does anyone have any questions so far on this slide or other slides or simulations that you want to ask? Okay. So in your final project, I always recommend, you can try to think to input. I mean, the final project, you will need to build or to invent a kind of a lab on chip. So it's always good to take a specific part of it, such as fluidics or electrochemistry, and try to build a model that simulates the behavior of this part, such as the flow or the signal, the chemical signal. Okay, so here we finished the first part of the lecture, and we talked about modeling and simulation of a lab on a chip. We talked about the key parameters. What are the steps that you should do when you want to build a model? First part is to ask yourself, do we really need a model? Yes, if we really need to build a model, something we can answer without, we can do by experiments, and how we can analyze the performance of the simulation to see that we indeed get the results or good results and not the bad results, such as garbage in, garbage out. Okay, so now we look into another type. Okay, so far we discussed about a very important part on modeling is taking your system, modeling the performance, building modeling, and to know and simulating your system to know how it works. Okay, you usually use modeling before you even go into building the actual device, because you want to see if your dimensions are good and obviously save a lot of time before you go directly into actually building or fabricating your device. Now, another important part of modeling is modeling the signal that generated from the lab on a chip. And for that, we talk about some kind of advanced modeling applications. And we define it as chemometrics. And chemometrics is a specific area in analytical chemistry. It focuses on machine learning or artificial intelligence algorithms that are being used to understand the signal that is measured or to extract chemical information or chemical information about the chemicals you have in the solution or the analytes. So here we try to identify first the components, and here I'm going to show an example of devices that can generate this kind of signals that they use for chemometrics, such as a multi-sensor array system. And then I'm going to show you how to analyze the actual work or the performance of these sensor arrays. I'm going to see some examples. So when we look so far, I discussed with you about specific receptors that are directly selective to the analytes. For example, we have the lock and key approach, which meaning that you have receptors that are very specific to one type of analyte. See the triangles. It won't fit the other types of analytes. Meaning that the more analyte will bind, the higher signal we get. And then we can have a regression model, a kind of like a correlation between the signal and the concentration. And we have a very nice calibration curve. And you can say, okay, this is if we are in this range, it's very high risk, low risk, and very low risk. Depending on the range, the number that you get. This is called a selective sensing. And we showed it in examples of, during this lecture, during this course. Now, there is another type of sensing which is based on cross-reactive approach. And here, the approach is actually not using a very specific receptors. We use partially selective receptors that, meaning that they are partially selective because they are selective also to other analytes in a solution. For example, we use a receptor partially selective, semi-selective receptor 1 that can bind to 2 different analytes, the red and the purple. Now we have receptor number 2, which also is not specific to a specific analyte, but it can bind to those kind of analytes and not, for example, to the purple. And the third one, the same also, it binds to this kind of analytes, but not to the red. So they are cross-reactive. They have overlapping in their sense, in the selectivity. And now what happens is that the response that we get from each one of them is different. So if we have a bunch of analytes here, okay, the same sample, sensor number 1 will get this kind of signal. Sensor number 2 will get another type of signal, and sensor number 3 will give a different signal. And all of these signals come from the same sample composition. But now we look from the from the top look at these kind of sensors, and we look at all these kind of sensors together, and we see a pattern. A pattern that means that when we have this kind of composition of a sample, we'll get this kind of pattern. This one gives you a lot of signal, less, and a bit more. And we can use pattern recognition algorithms to build classification or to train a model to identify specific fingerprints for a sample that comes from a healthy person, or a fingerprint that comes from a sick person. And we can build or train this kind of model or pattern to recognize that now, when we bring a sample that we don't know what we have inside, it can give us, and when we apply the model, we measure to get a signal, and we apply the model, we can give us a good estimation on if it's either the sample come from a healthy or from a sick person. So we look here and see 2 different approaches. The one which is the very selective and specific, the lock and key approach, or the cross-reactive approach when you have an array of sensors that each one responds differently and overlapping way to the other sensors. Let me focus on the second example. Does anyone have any questions on these 2 different sensing approaches?
Yes, I want to ask one question. I want to ask you how to classification training.
Oh, okay. How the classification training is going, and hold this question, and I'm going to show that in the next slides.
Oh, okay.
Okay. Any more questions on this slide? Okay. So first, let's understand where it came from, this kind of cross-reactive systems. And here, there are 2 main systems, biological systems, that inspire the approach of the multi-sensor array. There is the olfactory system, the way we smell different types, smell flowers or so forth. And we have another system, the gustatory system, which how we taste different flavors. And I have to say that I don't know if Weber is there, but Weber's work was mostly when it was in Israel, was on the flactory system under the supervision of Professor Hossam Hayek on Technion. And if we talk about my work, I work specifically on this kind of approach. So the flactory system is an example of how to measure molecules in the air. And the gustatory system is how to measure molecules in solution. And we take the machine gustation system and we look how, and I give an example, I give more examples, and you will forgive me because I come from the area of the solution detection, and I will give examples of how this kind of tongue approach is working, and we call it the electronic tongue. And the idea that we have an array of multiple sensors, that generate, each one of them generate different signals, and here it's kind of look like the electrochemistry signal. We do some kind of signal pre-processing, and then we use a chemometric model, we train it to with the data to get a decision results, if it either can be qualitative analysis, qualitative I mean a classification, or quantitative analysis gives us a reaction number. Now, before I go into this kind of tongue approach, how it works, we have also other types of senses. It's based on the human senses, such as eye or vision, hearing. This is more related to the olfactory system, how we smell, or how we taste, or how we touch. And the same work I'm going to show you here on the tongue approach can be also applied for other ways. For example, for multi-sensor arrays, when we use, such as for examples like eyes working, we can use an array of cameras to generate multi-sensor array and to use it with the machine learning algorithms. Or an array such as the ear works on sound, we can have an array of microphones. Or touch, or smell, and this is the electronic nose that I was telling about, or the taste, which is the electronic tongue approach. So any questions so far on the general level of this kind of multi-sensor arrays? Okay, not okay? Okay. So how it does actually work, and I give an example before I go to the actual examples, the actual working principle. This is an example of a paper that uses optoelectronic nodes, meaning it has an array of colorimetric or optical sensors that each one has a different type of chemical inside. And now when each one is an optical sensor, has another type of chemical that reacts with the analyte. Here, for example, it reacts with N-butylamine. We add N-butylamine to each one of the sensors, and based on the reaction, chemical reaction, the color changes. And it differently changes because in each chamber you have another type of a chemical or reactant or cofactor. So when we start to test the response of the sensor array to different types of chemicals, we start to see a pattern. For example, for N-butylamine, we don't have any reaction from this chamber, while for pyrrolidine, we do have some kind of reaction in this sensor. And then we need to generate this kind of patterns from the different molecules that we want to measure. And we use this as the input to train an algorithm that will recognize these kind of different patterns. So how can you extract the chemical information? And here is the question that is more related to what you asked earlier, to how to train those models. So there are different, there are different names. There are chemometrics, there are machine learning algorithms, there are artificial intelligence, and multivariate mathematical models. And the idea is that all of them had the same idea, that you have a training data. You need to generate a training data, such as here was measurements or see the colors that are generated from different molecules or different combinations of molecules, but you need to generate a lot of training data. Then you teach the algorithm. You use the training data to develop a model that will predict or will explain this kind of training data. And then you can test the algorithm that you developed on another type of data that you didn't use for the training, and use the test data and see if on your model, and see if the answer that you got is indeed similar to what you have in the test data. There are different types of geometric models. We can categorize them into two different two different categories. The unsupervised learning, meaning that we don't have the part that we actually teach the algorithm. We can actually just apply the training, use the data itself and get the answer because of some kind of variability that we measure in the data. And there is a supervised learning, which is the one that you indeed use the training data to teach the model. So supervised and unsupervised. Now I won't go, this by itself, it's a whole course, and actually I have another course called Bioelectrochemistry that I teach these kind of approaches more in depth. But anyone that is interested in learn more, first you can send me an email, or even and I can recommend some books, or also can talk to me about kind of maybe internships, or can come to my lab and learn more about this kind of approaches. So we have the unsupervised learning, which give us clusters information, so we don't need to train the model. The model gives us structures inside the data, identify different structures, so it gives us some kind of clusters in the data. Or we can also use this kind of approaches to reduce the dimension of the data. Usually we generate a large matrix of data, which most of that is kind of redundant, so we can decrease the dimensionality of the data. Here I put some examples, and the most common one is PCA, partial component analysis, and I will give an example of how to use it. So that was the unsupervised. When we don't train our data. We have the supervised learning, that we actually train a model based on the data. And here you can have two different types of model: the regression model, when we predict specific numbers on the data, such as concentration, or classification, when we predict different classes, healthy, sick, and so forth. And we can use SVM, ANN, and so forth. Any questions on this slide? Okay. Here comes some, gave some examples. There are many different types of models. There are for example, quantitative, supervised, multilinear regression or partially square, pattern analysis, the unsupervised principal component analysis, or supervised principal component regression. There is the known one, which is biologically inspired, such as the ANN or fuzzy methods and so forth. It's a whole field by itself. Here I gave you the acronyms for each one of them, and the most used one, the most used one is principal component analysis and partially square. There are some examples of ANN, artificial neural network, but these are the main ones that are being used currently in the field. Now I want to focus on partial principal component analysis, the PCA, and to give us an example of how it actually works. And PCA is based on that is based on the principle that the data that we have has the way that we present it, which actually in the real world, such as concentration or signal, specific signal that we measure as a function of different concentrations of the analyte that we have, it's actually, there is a better way to show the variability in the data. And the example is this kind of graph. So let's take an example that, for example, this is the signal that we measure, I don't know, like the intensity or the absorbance of some kind of in specific wavelengths. And this is the concentration of our analyte. Oh, no, it's not good. This is the log the concentration. Okay. Okay, sorry. Now, when we look at this kind of data, you know, you show, and when you look at the variability or the variation, my arrow disappeared. Okay, no. Draw. No. Yeah. So when we look at this, for example, for this, we look at this point and this point, the variability or the distance between the two of them is going to be this kind of distance. So we have some kind of variability or some kind of sensitivity that you can differentiate between the two of them. However, there is another set of axes of coordinates that will give us better difference. And this is what PCA is doing. PCA, principal component analysis, finds those kind of coordinates that maximize the variability. For example, you can find that this kind of axis will actually give you better variability, because now when you look at those two samples, they are here and they are here. And this is much better or much bigger distance than this one. So you find a new coordination that gives better distance or higher variability between the points, the data points that we measure, than the one that we used originally. And we call this kind of new coordinates as principal components. So the PC, principal component 1, will be the one with the highest variability. The second one will be the one with the second most best variability, and so forth. You can go into 3 dimensions, and so forth, actually to 4 and 5 and 6 and n dimensions. And they are all orthogonal to each other. Does anyone have any questions on PCA? Okay. So the whole idea is that we take our data and we look at it from a different point of view that will maximize the variability. So when you apply PCA, and you can look at it online, it's actually very simple. It's based on specific some linear algebra that you build a correlation matrix and you do a transform to find the most variable axes. So this is an example of an unsupervised learning that we just put the data, and now we did this kind of transform that give us a better axis to look at much higher variability between the data. We didn't need to do any training. Now there is a supervised learning that again, that we use and we teach. This is the training data to recognize specific information. And then, for example, we use this kind of unknown or test data to see on the model, and it gives us information, okay, based on this model, what we have here in the image is a cat or a dog. So this is the same example with the supervised learning when we use on the multi-sensor array that we develop a model and we train a model, and then we use the test data to test how accurate our model is. So any questions so far? And I can say that the best way to understand this kind of subject is to try by yourself with your own data to build a very, very simple model such as PCA and see how it works. So the one that the multi-sensor array that I will focus on is the electronic tongue. And this is the area that I'm working on. And the reason I'm focusing on that because it can be used for different applications, such as safety, to recognize specific hazardous molecules in our water, to recognize specific food products, if it's either the quality or the origin, or even the taste eventually of the food. You know, we can train algorithms to know the taste of the food. We can use models to analyze pharmaceuticals if we have any what is the actual concentration of the pharmaceuticals in our drugs, or using that in the biomedical field to recognize specific or analyze biological fluids and recognize specific medical conditions. Electronic tongue is very, very sensitive. You actually, in most cases, you need a very small number or even none to sample preparation steps. You don't need to do any separations or filtering. It gives a very rapid and cost-effective detection. Because we build on the variability between the sensors based on the sample that we have, we actually decrease dramatically the effect of interferences. Because if we have the same interference on each one of the sensors, it won't contribute to the variability. So we can so eventually when we do an analysis, the model will be mostly about the actual chemicals or analysis we have in the sample and not the interferences. And very nicely, we can actually use the same signal and build models. This kind of models can actually predict for multiple molecules or different multiple analytes. Such as we saw before, that the sensor was able to detect different types of analytes, not only one of them. Any questions so far on this slide? So how it's being used, how it looks like. Electrochemical electronic tongue is based on an array of electrodes, give us the array of sensors. And here is an example, and we have a multiplexer of different working electrodes. We have a reference electrode. And for example, here we measure the potential change from each one of the electrodes, or we can also measure the current from each one of them. And this is a benchtop system that that you have multiple samples, and this is a sensor and measures from each one of them to generate a large data set to train the model. You can also miniaturize those kind of sensor arrays. Is an example of these 3 different working electrodes made of different metals. Each one will generate, if you put the sample on top of it, each one will generate a different signal. So you have an array of 3 electrodes. Usually, the detection principle, the detection technique that we mostly use in electrochemistry is potentiometric. If you remember, there are 3 different detection methods: the potentiometric, voltammetric, and impedometric. We learned last week. So most of them are potentiometric. There's some voltammetric, measure the current, or impedometric, measuring the conductance. And these are the non-electrochemical ones, such as colorimetric or mechanical and so forth. Number 3 is paper. The main application, okay, in econometrics, what kind of tools or models are being used? The most one is PCA, as I told, but also LDA, linear discriminant analysis. It's a kind of a classification model. Next to it, we have ANN, artificial neural network, and partially square regression or classification. And then we have others which much lower number of applications. Now, when we look at the input data, we can actually think about the way that we measure the signal, because the way that we also measure the signal, we also contribute to the variability, such as if we apply the potential, and I think actually like a linear scan, we apply the potential and we measure the, sorry, we apply the potential over time and we measure the current, such as linear scan voltammetry, or we use chronoperimetry, which is when you apply a fixed potential and measure the current. So these 2 kind of techniques will also contribute to the variability in the signal. And it's very important that we want to increase the variability between the sensors, remember, because then it will give us more information about what we have inside. So the example I want to show you, first example of a voltammetric electronic tongue, is from this group. In this paper, they had different types of honeys, sweet honey that comes from bees, and they wanted to build a model that can classify or tell you from where this honey came from. based on voltammetric electronic tank or the electrochemical signal you measure from each honey. So first is generate the data, and they chose different types of honeys from different locations, Germany, Canada, Morocco. So they generate a data set from different locations, from different types of honey. And this is their setup, which was a portable electrochemical system, a laptop, and a potentiostat connected to a multiplexer because you want to measure from different electrodes. And they put the kind of electrodes array in the sample and they measure the signal. So this was their setup and their samples. This is an example of how the signals look like. This is an electrochemical signal. And this is an example of a single electrode, how it measures the signal from different types of honeys. From GH1 or pH and CH1, these are different types of honeys, different groups. And this is the potential, this is the current was measured, they're changing the potential and measuring the current. What we can see that overall it gave the same trend, like increasing the potential you get higher currents, but there is some still variability in the signal. When you measure this kind of honey, GH1, honey that came from West Germany, compared to pH, honey that came from, from Portugal, we can see that the current, maximum current, here was 4 microampere, here was 8 microampere. So something happened there, something in the honey, some molecules that are being oxidized that the one from Germany had much higher concentration than in Portugal, and so forth. So we look at the signal and it's a bit different from each origin, from each type of country. Now there is the pre-processing, the data processing step is that we don't want to take the whole signal. Let's take features. It's called feature extraction. We took features in the signal that contribute mostly to the difference between the different types of the honey. So what they took is those 3 different features. The first one was delta I, which was the difference between the current at the maximum place minus the current at the minimum place. This is feature 1. The next feature was S oxidized, meaning the slope. This is the oxidation current. So the slope, the highest slope at the oxidation area, and the highest slope at the reduction area. So they take those 3 different parameters, they take the data, they do the data processing, they extract these 3 different values for each electrode, and then they took all the data and put it in a PCA, applied the PCA algorithm in an unsupervised. First, I want to see all the data to see if there is some kind of clusters or structure in the data. And what they've seen, now remember the PC1, PC2, PC3. And what they observe is that there is some kind of different contribution to the variability from the different groups. For example, if you look at the pH, the one that came from Portugal, it has a different cluster than the one that came from Germany. And also from the other came from different countries or different types of trees, of flowers. So what they can see that even though they look the same, more or less, by extracting those 3 different features, values from each one of the electrodes, from each one of the honeys, we can get a very good clustering or differentiation from the different types of the honey. So this is an example of, it's a very, very simple one of how you measure. You measure the chemical signals, then you analyze, you do some kind of pre-processing to extract the features, and then you apply an algorithm. Here was unsupervised algorithm to see if you can have any structures or if you can differentiate between the samples. So do you have any questions on this example?
I want to ask, for the sample of the NH, does it mean it isn't good enough for the classification?
You mean if this kind of different clusters, if it's good enough for classification?
Yes, for example, the NH sample, the green.
Okay, so in order to classify, so basically right now we didn't build any model yet. Okay, we didn't build any training, we didn't train any model or classifier, meaning that, okay, now we bring another type of honey that we don't know where it's, what was its origin, and we measure it and we see, for example, if it falls here. So we can kind of qualitatively say, okay, from the figure, I think it's related, it's the one from Germany. Or it falls here, it's the one from Portugal. But we don't have yet, we need to look at it, and it's not the model that I was talking about, the classification model, meaning that the model that you add the values, it calculates it and get you the answer. For that, we need to use, take all this data and build classification models. So it means that for PCA, the classification model called linear, or the most common one is linear discriminant analysis, LDA, meaning that it finds kind of planes, you apply it and it finds planes that differentiate the clusters from each other. So for example, it finds a line between this cluster and this cluster, a plane from this cluster and this cluster that will be kind of the highest or the same length from both of them. And so forth, it does it for all the other clusters. So it calculates planes, and now it gives you basically on the data that you, or this is the model that you trained, and a lot of data gives you a different planes for the different data, just like this, and so forth. Oops. And now when you, this is the model that you build, you know, discard this kind of planes in numbers, in mathematical equations. And now when you have your data, don't know where it came from, it falls here, for example. And now from all this kind of, from the model, it tells, okay, there are chances, the most chances that is related is here. So it tells you, and you build a thing called a confusion matrix. It tells you, okay, we put a sample, let's go GH. And we take some samples from the Portugal ones. GH and PH. So we took a sample from the G, and we know it's G, but the model tells us, yes, it's from G. So it's correct. So you get 1 point here. If the model tells us no, I think it's from Portugal, although it's from G, so it falls here. It's mismatch. Same thing for a sample that we give from Portugal, and we get a good prediction, so it will fall here. But if it's sample from Portugal and tells us no, it's from Germany, it falls here. So that's why we take multiple test samples until we get a confusion matrix. For example, we took 10 samples, and 7 of them were correct. Tells us it was it was the one that were for Germany were from Germany. 3 of them were for Portugal. I said Portugal, but there are another. Okay, took 20 samples, and 5 of them said. Sorry, 10 of them were wrong. So what tells us the accuracy is this part, and then we calculate the accuracy or the sensitivity of the matrix, and these are the numbers that tell us how good our model is. So this is the classification model. I haven't gone, it was only like 1 or 2 minutes. But this is the way that's being done, and also a similar way for regression models. Like if you want to know the concentration. So not the origin here, concentration, how much we have from specific molecules, it's called regression models, and this is a different way to do that. Okay. So this is how you build a classification model. Only for 1 minute or 2 minutes, but if you want to learn more, let me know and I will send you more information on that. Okay?
Okay, thank you.
Sure. Any questions on that? Very good question, thank you. Another example I'm gonna show is, it's an actual paper that here they wanted not to know origin of of materials. They wanted to know the concentration of catecholamine neurotransmitters. Neurotransmitters that are molecules that transmit information to our neurons, and they wanted to know the concentration of these kind of neurotransmitters in urine. And they used for that electronic tank based on not electrochemical, but based on optical sensor. For that, they have 3 different sensors that generate color. And they saw that for example, for L-DOPA, it's a kind of neurotransmitters, you get a different pattern. For dopamine, you get another pattern. And for norepinephrine, they tested 3 different types of neurotransmitters, you get 3 different patterns. And they also tested, okay, now we want to have a mixture of both norepinephrine and L-DOPA, and they have a different pattern. So but what basically they show, they have 3 different sensors, and they show the 3 different patterns for 3 different neurotransmitters. And the sensors are actually quantum dots, meaning that these quantum dots, that whenever they interact with the catecholamines, catecholamine is general name for different neurotransmitters. The catecholamine, they change the absorbance of the color. So they have different absorbance for different colors. And this is how the absorbance looks like, the emission, how much the emission of light for each one of the quantum dots. So for quantum dot number 1, type 1, this is the pattern that measures when you have norepinephrine. This is the pattern when you don't have anything, which is the blue. And the green one is similar, which when you have L-DOPA. And the pattern changes when you have dopamine. So what you can see that it's a partially selective sensor. This is a sensor because it reacts differently to each one of the analytes. This is sensor number 2, a different type of a quantum dot. And again, we can see the different signal for each one of the molecules. And this is the third one. So this is how we validate first that we generate different signal for each one of the molecules that you want to measure from the 3 different sensors. Now they generated data and they measured different concentrations of norepinephrine, L-DOPA, and dopamine from each one of the sensors. And they put all the data in PCA. Remember, PCA want to find a new type of coordinates to maximize the variability in the signal. So this is how it looks like, and they showed PC1 and PC2. And by the way, these numbers next to the PC1, this is the percentage of the variability that PC1 shows. And this is the percent variability of the PC2. So to say that the PC1 has the highest, represents the highest variability in the signal, PC2 is the second level, and third, and fourth, and so forth. So let's look at the data itself. In norepinephrine, these are numbers at different concentration, increasing concentration of norepinephrine, and we can see the cluster is here. And obviously the point changes because you have different amount of norepinephrine. Same for L-DOPA and same for dopamine. So it means that the signal measured from those 3 different sensors, when you integrate them, you have enough variability to represent different clusters for the different molecules. Questions so far on this slide? It's clear how the data was done. Like first you extract the emission from each one of the sensors from different mixtures, and then you utilize PCA to see if there are any variability in the signal that you can represent differently in a more clear way. Now they said, okay, let's try and add some kind of mixtures of norepinephrine with L-DOPA or L-DOPA with dopamine. And what they saw that when they add norepinephrine with L-DOPA, this is where the cluster falls. This is where the sample falls. It actually makes sense because if this is only norepinephrine and this is only L-DOPA, when you have a mixture that you have both of them, you have some kind of in the middle because the variability is contributed by both norepinephrine and L-DOPA. So it falls in the middle. Same for L-DOPA and dopamine, or norepinephrine and dopamine. So it's a way to tell us if we have a mixture and we have the sample that falls around here, so we can say, okay, we have a mixture of both dopamine and L-DOPA, or if it falls here, we can say, okay, this is norepinephrine or L-DOPA or dopamine. Any questions on this slide or on this paper? Any question in general on advanced signaling processing? Okay, so this is a field that increasing exponentially, and I very much recommend you to learn about it more. And always, feel free to ask me any questions. So here we finish the second part of this lecture on how we can use chemometric or artificial intelligence-based models to interpret multi-sensor array systems. And here I'm finishing the lecture. It's a bit early, okay, I'm 10 minutes, but I'm finishing the lecture because tomorrow it's gonna be our last lecture that we're gonna talk and understand, okay, we talk in theory about the different components that we have, how to build in theory the device, to model it, and so forth. Now, tomorrow we're gonna learn actually how to build it, how to fabricate it from scratch. Okay, so if you don't have any more questions, I can say goodbye and see you tomorrow.
Bye.
Bye-bye.